Okay. All right, everybody, let us start. Please have a seat. Okay, have a seat, settle down, please. Thank you. Okay, today's pitch, I always get emails asking me to pitch things to students, and I will do so, and you can make of that, attend or not attend, whatever you want. Today's pitch is the Women in Science Research Conference this coming Saturday. We're invited to join on Zoom and learn about research positions, how to find research positions, and how to improve your resume. There will be professors, grad students, and undergrad students there, so it should be a good mingle. All right, it's a very exciting week for parents this week. I don't know if you guys have any kids at home or younger siblings, cousins, nephews, nieces. I'm very excited that I will hopefully get some of my kids vaccinated this week. So that'll make me a little bit less paranoid of being in a big audience with a lot of students, because I can still carry it home to my kids, and I would like to avoid that. So very exciting for us. All right, so today's topic in biochemistry, because that's what we're here for, is RNA processing. So last week we had talked about and finished with how RNA is made and how RNA transcription is regulated. So the question is, what happens with the RNA after it's being made? And there are several steps that happen in eukaryotes, not so much in bacteria. So in bacteria, I will try to talk slowly. I've gotten requests via email to please talk more slowly, but please always feel free to just raise your hand, let me know. I'm not doing this out of malintent. I really want you guys to be able to follow along and understand what I'm talking about. So if you just want to, raise your hand, but like slow down, please, that'll give me a good reminder to do so. Okay, that being said, in bacteria, the transcription happens in the cytosol. So the bacteria don't really have the structure of the nucleus and then the cytosol for translation. So in bacteria, it all happens at the same time, which is also cool. So in bacteria, as soon as your RNA is made, your RNA is also starting to be translated into proteins. So we always, think of it as a okay, first we have the DNA, now we make the RNA, then we make the protein. It actually all happens at the same time. And I know some of you have already noticed in the additional lecture material, there's the TRP operon, which is a regulatory mechanism that happens when transcription and translation happen at the same time. I will not be asking about this in the finals. It's just a really, we used to have it in these lectures, which is why we keep it in case we want to put it in again next year. But it's a really cool mechanism on how bacteria, by coupling transcription and translation, can control what's in their cells. But again, not being tested. Okay, don't have to worry about it. In general, everything that I will test on the finals will be covered in the lectures. That doesn't mean you can completely ignore the book. Okay, so if you don't understand something, if you want clarification, if you want to learn a little bit more about it, and that helps you understand it better, then please look at the book and the lecture, the additional material that we post online. This is supposed to help you understand things better. I will go over everything here, but sometimes, I might just have it on one slide and not covered on 10 different slides. So, pay attention to what I cover here and then look it up in the book and look it up in the additional material. Okay, so the RNA processing topic is really about what happens in eukaryotes, okay? So in eukaryotes, the RNA transcription happens in the nucleus, and then translation into protein happens in the cytosol, which gives the cell, the time to process RNA, okay? So it can make changes to the RNA, while bacteria can't do that. It already translates, so it doesn't have time to process it. Here are your objectives, and this is the first section, RNA processes. So again, RNA processing happens in eukaryotic cells. The pre-mRNA is the RNA that's first transcribed from the genome. Okay, so when you have your DNA and you make the first RNA, just after it comes off the polymerase, that's called the pre-mRNA. The mature mRNA is the one That's then the messenger mRNA that gets translated into proteins. So pre-mRNA being processed into mRNA. We have three main things that happen, plus a couple minor things. So we have the 5' end gets capped, the 3' end gets polyadenylated, and introns are being spliced out of the mRNA. Okay, three major things happening in the cell. Okay, so this is your pre-mRNA here. First, you have the DNA that's being transcribed into the primary transcript. The 5' end then gets a cap structure. The 3' end gets a little bit truncated and then gets a poly-A tail added to it. So just a lot of adenines in the end here. And the introns are being spliced out. So intersecting RNAs that we don't need for the protein coding frame are being spliced out. All of these things, especially the capping and the poly-A tail, are meant to make the RNA more stable. So as I mentioned before, RNA, when you have it in the test tube, is super unstable. The same happens in the cell. RNA just isn't, that's just biochemically is not very stable, but also in the cell, there's those enzymes called RNases and exonucleases and endonucleases who will chop up the RNA as soon as it's unprotected. So if an RNA is unprotected in the cell, it will get degraded, which is part of, okay, so you best know, right? The vaccine is an mRNA. So we're putting an mRNA into our body. The first problem is to deliver it into the cells, but then also to make it stable enough that it actually gets translated into proteins. It isn't stable very long. So after 24 hours, that mRNA that they give you, that they vaccinate you with, is going to be gone. What is left is the protein that's being made of this mRNA. Which is why when people say, "Oh, it's gene therapy, it's going to edit your genome," none of that can happen, and it gets degraded fairly quickly. I can tell you, we try to isolate mRNAs. They are unstable. You have to be super careful. They always get degraded, and it's a pain in the lab to work with those things. So they're not easy to work with because they're unstable. But the cell has those mechanisms to make them more stable in the end. Okay. So let's start with the capping. The capping is the addition of a reversed nucleotide, basically. It's a methylated G nucleotide, and you can see it here. This is the regular G from GTP, right? And it has a methyl group up here. You do not need to be able to draw this, okay? I will not ask you to draw structures. You should know that it's a methylated nucleotide. And then what's really special about it, if you look at this regular base here, this regular polynucleotide, you have a 1-phosphate group here, right? Just one in between those. You can see these ones, there's 3 phosphate groups in between, and it's not on the same position. So it goes from here to here instead of here to here. So instead of having 5' to 3' to 5, can't talk, 5' to 3', you have a reversed nucleotide addition here. What this means is in the cell, this cap stays on until a very specific enzyme comes to cleave it off. So the regular RNA degrading enzymes that can just chop up the RNA, they can only chop up this regular bond here, the regular 5' to 3' bond. They cannot chop this one up. So there's a specific decapping enzyme that can take this cap off. But what that means is your 5' end is protected against degradation by having a nucleotide just in the reverse order on it. And then again, the methyl cap here, the methyl group has kind of the same function. It makes it look a little bit different so that it can't be recognized by the regular RNases. Okay, so making it look different makes it more protected against degradation. Does that make sense? Probably? Okay, I'll assume you'll understand unless I hear otherwise, all right? Okay. It's kind of like my kids at school. I assume they're doing well until the teachers tell me, but it doesn't always work that way. All right, so 3' end. The 3' end also gets protected. There's no special bonds here, so it's a regular 5' to 3' nucleotide addition that we have here. It's mainly the amount of bases that we add here. So we have our regular RNA, and then we have up to 300 bases. Sometimes you can have even 500 bases of adenines added to this tail on the 3' end. So this is a regular polymerase. The difference is that it adds the nucleotides untemplated. So it doesn't require a template. There's no pulley T tail in the DNA that tells the RNA polymerase to add the A's here, okay? So you have a long A tail here. And now you can think, well, it's the regular 3' end in the end. How can that protect the RNA from degradation? It's not just the length of the A's. It's also proteins that can bind to those long A stretches. So there's something called a poly A binding protein that binds to those A stretches. It doesn't bind to a regular RNA, but it can bind to those long poly A tails. So the longer your poly A tail is, the more poly A binding proteins you have bound to it, the more stable your RNA is. Now the kind of disadvantages every time you translate an RNA into a protein, the poly A binding proteins get somewhat displaced for a little bit, and then the RNA gets shortened. Okay, so over the lifetime of an RNA, the RNA gets shortened, and once the poly A tail reaches a certain shortness or length, the RNA gets degraded. But the length of the poly A tail is what determines the lifespan of an RNA, unless there are other signals that tell us that the RNA isn't needed anymore. Okay, so that's your poly A tail. When you have your primary transcript here, your pre-mRNA, this is your 5' end, it gets capped, as I mentioned. And here's a little bit of an untranslated region. Somewhere here, we might have the start codon where translation starts, and we'll talk about translation in the following lecture, so you don't need to know about that now. But at some point, the protein is done, and we have what's called a stop codon. And then after the stop codon, that RNA is not really needed anymore for the purpose of making a protein. So after that stop codon, which is the last codon after the last amino acid, we have a consensus sequence, which we've learned about before. In this case, it's an AAU AAA. Again, you don't need to memorize consensus sequences. And after that, you have a little bit longer transcript here that the RNA polymerase makes. Okay, so this is your primary RNA transcript. Then the poly A polymerase comes in and recognizes this signal here. It then cleaves the pre-mRNA right after this AAU AAA sequence. Which means this section here that you originally have in the pre-mRNA transcript is not in your final mRNA transcript anymore. And after the signal, it adds about 300 bases of adenines. And then those bases are being bound by the poly A binding protein. So your poly A polymerase, your poly A polymerase has several functions. It can recognize the sequence. It has an endonuclease activity, so it can cleave the RNA. Okay, it can't just add RNA, it can also cleave it. It cleaves the RNA and then it adds all those adenine residues. Okay, so 3 functions: recognizing, cleaving, polyadenylating. Okay, the importance of those tails are, and the cap are, they mark the 5' and 3' end as being intact. So sometimes an mRNA is not being made properly, the polymerase falls off too early, doesn't get a poly A tail. In that case, the cell really doesn't want the mRNA, so that our mRNA would get degraded right away. So an mRNA that has the 5' methyl group and the poly A tail is being marked as intact. These, both the cap and the poly A tail, are required to export the mRNA into the cytosol. Okay, so far we're still in the nucleus. We've made our mRNA, and now we're processing it so it can be exported to the cytosol for translation. And then in the end, they protect the mRNA from degradation. Okay, so the mRNA can be degraded in both the nucleus or the cytosol, and it needs both the cap and the tail to not be degraded. Okay, which gets us to the last modification and likely the most complicated one, which is RNA splicing. In bacteria, the mRNA is being made in the sequence that it will be translated. So in bacteria, when you look at the gene sequence on the DNA level, you can already see what kind of protein that makes. In eukaryotes, we have hugely long pre-mRNAs that are then being condensed to shorter mRNAs. Okay, so the pre-mRNA is much longer than what we need in the end to make into a protein. they have what's called introns. So the coding regions in an mRNA are called the exons, and the non-coding regions are called the introns, and they interrupt the sequence and have to be taken out before we go into translation. So these introns are spliced out of the primary transcript to make a mature mRNA. So what happens is you have exon 1, exon 2, and exon 3, and then you have those intron sequences. And then we have a cut here and a cut here, and then they're getting ligated together to form the mature mRNA. And only this mRNA then makes sense in terms of being a protein coding sequence. So if you were trying to translate this sequence, you wouldn't get very far. Okay, we'll talk about why frames are important and how mRNA is translated into proteins in the next lecture, but you have to splice the introns out. So if the introns aren't taken out properly, the RNA is not going to be functional. And that's, so we sometimes in cancer causing genes, we have mutations in the splicing sites, and then the proteins aren't being made properly, which can lead to problems further down. Okay, so exons and introns, an easy way to remember this: exons are expressed, introns are interruptions. Okay, so you have to know what exons and introns are. You need to know those definitions, because that's really very basic knowledge. Now, the pre-mRNA of the pre-mRNA that's first transcribed can be huge. So this is a pre-mRNA of the human factor VIII gene. Okay, just a human gene. And it's 200,000 nucleotide pairs long. That's your pre-mRNA. And then it gets condensed to an mRNA that's only 2,000 nucleotide pairs long. So you can see eukaryotes are wasteful. Remember that I told you guys that in bacteria, everything is condensed because they have to be fast at replicating their genome. That selection pressure does not apply to eukaryotes, and they make those huge genes where most of it is spliced out. Now, we used to think that anything that's spliced out is not needed and not being used for anything. Now we know that's not true. So some of those introns that are being taken out actually have other functions for regulating totally different genes. You can even have tRNAs encoded in those genes. So some of them have functions, but still most of it is just waste. So it's really incredible how wasteful they are, or we are as eukaryotes, because we make all those, it costs money. It costs a lot of money. If you remember how all those proteins have to come together, how they use ATP, costs a lot of money, money in terms of cellular money, currencies, to make those pre-mRNAs, and then take out most of them. But you can also see that it's a lot of different exons that have to be spliced together, and all of this has to be done very correctly for it to work, to give a functional protein coding gene in the end. Okay, why splice? What splicing does is, despite being wasteful, it also increases the coding capacity of the genome. And eukaryotes can do something that's called differential splicing, where they can splice things together in different ways. So if we go back to this figure, you always start here with number 1, but you can then use some of the exons, but not all of the exons, to make different forms of the same protein. So you can use the same mRNA to have different coding mRNAs in the end. Okay, so same pre-mRNA, different mRNA products, different protein products in the end. One thing to remember is, even though you can skip certain exons, you can never go out of order. So I can't start with 1, then put in 22, then go back to 5. Okay, so you can leave them out, but they're always in the same order. Okay, so there's no jumbling it around. Okay, this would be something that would be a suitable exam question, basically. Assuming the 5' most exon is always expressed, which, as for the sake of this course it is, and splicing occurs in a linear fashion, meaning you can leave out exons, but you can't go back. So you can't go 1, 3, 2. How many protein products could be made from the following gene? Okay, I'll give you some time to figure that out, and then we can do raise hands, or I can walk you through it, whatever you prefer. Okay, do you have the answer? Yeah, you got it. Okay, I'll repeat it so that everybody can hear. The first combination would be all 3 exons, 1, 2, 3, all spliced together. Next combination is we just don't like exon 3, so we'll take exons 1 and 2. Another combination would be exons 1 and 3. That's now your third version of the same protein. And then the last one is you only do exon 1. You don't go further than that. So out of one gene or one pre-mRNA, you can make 3 different ones. Yes, question? Okay, so the question is, why is it not possible to just do 2 and 3? You need, if you remember, you need this whole RNA polymerase binding site, right, in the beginning to initiate transcription. If you had another transcription start site here, then you could do 2 and 3. But assuming that we only have this right here in the front would mean we have to have exon 1. Now I can tell you that it's a really good question because we actually do have different transcription start sites where we start here, but they're rare. So in general, we can assume that exon 1 is always expressed, and in an exam question, it would specify that exon 1, assuming exon 1 is always expressed. Good, understandable. More question back there. Yep. Okay, yeah, you can have, I think the question was if you could have just 1 and 3. Yes, you can. I think that was it. Okay. Does that make sense? How you can have different gene products from the same gene? Okay. Now this is an example. This is a muscle gene, okay? Alpha tryptomyosin gene. It's just it's a protein product that's being used in all kinds of different muscles. And these are the different splicing variants here that we find in different kinds of muscles here. So striated muscle, smooth muscle, fibroblast mRNA has 2 different ones, or brain mRNA. So one protein is differentially expressed in different tissues. Usually, now this is a broad usually within the same cell type in the human body or in higher eukaryotes, you will have 1 splicing variant per tissue. Okay, so one tissue does this one, the other tissue does that one. It doesn't just hop back and forth between the different splicing variants. Okay, there's always exceptions to any rule that I'm going to teach you. Okay, so take everything I say with a grain of salt, which is why in multiple choice questions, choose the answer that's most correct. Okay, because exceptions to the rule still means 99% of the time the cell will follow the rules. So why would we have different versions here? Let's say, and this axon here in the very 3' end, let's say that axon directs the protein to the cell membrane. And only in these 3 cell types, the fibroblasts are the same here, only in this 3 will you need the protein at the membrane. So you can localize the protein by either having that exon or not having it. And here's your exception to the rule. You can see fibroblast mRNA has 2 different kinds where one time it has this exon, one time it doesn't have it. So another one you could see here, this is, you either have this one or this one. Okay, you don't have both exons. You either have exon 2 or exon 3 here. Let's say that means you either interact with protein A or you interact with protein B. But both interactions aren't needed in a specific cell type. So by usually those exons, they correlate the sequences that we splice out or in. They correlate with different functions like protein-protein interaction, localization, nucleotide binding domain, things like that. Okay, so those different exons, not always, but usually you can say, oh, this is what that exon does in this specific cell type. Okay, so this, even though this intron, having introns, it adds a lot of junk RNA. In the end, it adds a little bit more. It adds a layer of control about what the protein does in the end. Okay, so how does splicing work? And I'll try to walk you through this. Here are your consensus sequences for the 5' splice site, the 3' splice site, and the branch point. The one you have to remember is the A. It's called the A branch point, and it just has that name. It's in A. please, that's the only one I'm going to ask you to remember. Remember the A, okay? So remember that there's an A branch point, and then you should remember that there are consensus sequences here and here, okay? You don't need to remember what they are, but you should remember that the consensus sequences are there. And you can see part of this consensus sequence is in the exon and some is in the intron. And that's the same for both sides. Which also means after you're splicing, your finalized splice site is always an AGG. Make sense? Because you cut everything yellow out. You put the 5' exon and the 3' exon together, meaning you add up with a spliced together RNA that has an AG and a G. This branch point is going to be gone because it's in your intron. So the intron, the yellow section here, is the section that's being cut out. Okay. Okay, so this how it happens. Is 2 different reactions are needed to splice an RNA out, and so they are, very clearly explained here. But and basically, what happens? You have this branch point 2' OH group, and this 2' OH group attacks this upstream splice site here. Okay, and then this loops back and forms a lariat structure, a lasso structure, okay? This hydroxyl group here is now free, the 3' hydroxyl group, and attacks this splice site down here, which then allows the RNA to be ligated into a spliced RNA product. So for exams, I know that's like I'll try to really make clear what's important. Okay, so I'll try my best. So for example, remember the 2' hydroxyl group attacks the upstream splice site, forms the lariat structure. 3' hydroxyl group attacks the downstream splice site, and that leads to the formation of the mature RNA product. This is not just happening out of the blue. Okay, so there's a huge question. Sorry. Lariat structure, L-A-R-I-A-T. I might be pronouncing it wrong. I'm German after. I don't know. Lasso. It's a lasso. Okay, it's here. See it? Right here. Okay, so this is a crystal structure of the yeast spliceosome. So this is the protein RNA complex that recognizes the RNA, the pre-mRNA, and makes it does the different splicing reactions. So the cool thing about this big structure is that it actually has a huge RNA component. So it consists out of RNA and also consists out of protein. And the way how these ones are recognized here are your consensus sequences. In this case, actually not, we don't have protein DNA interactions here. It's not DNA, it's RNA. So the way how this is recognized is not actually by a protein, but it's by the RNA subunits of the spliceosome, which have sequences that are complementary to this sequence. So in the spliceosome, you have an RNA sequence that's reverse complement to this. So it would start U would base pair with an A, G would base pair with a C, A's would base pair with a U base pair with an A, and so on. So it's actually the RNA subunit of the spliceosome that recognizes the 2 sites that the spliceosome has to bind to. Okay, so your 5' and the 3' 5' and 3' splice sites are recognized by the RNA subunits of the spliceosome. The protein itself is, so here you can see in green and red are the RNA subunits, and then in gray are the protein subunits. And the protein subunits are mainly in here as a scaffolding protein. Okay, so they help recognize, but or help put everything in the right position, but the RNA is what recognizes these splice sites. Okay, this is just to repeat it one more time. Here you have your 5' exon, your 3' exon. You have your 2' OH group of the A branch point attacking your 5' splice site, forming of your lasso structure. Second attack forming the mature mRNA, and this one is released and degraded. Okay, so you get this. cell recognizes a circular RNA structure as not wanted and foreign, which is kind of the same what it does with viral RNAs are usually circular and are also recognized as not wanted and foreign unless they're really good and evade the RNases. So that's the first response of the cell to foreign RNA, is to just degrade it. Okay, again, alternative splicing, we already went over this, so I don't know why I put it in here again. You get the different combinations of proteins from the same pre-mRNA. Question, yeah? Yes, yes. So the splicing of the intron, what happens when you have, well, the question is whether the splicing of the intron determines the absence or presence of the exon. In order for an intron not to be recognized, you have to have mutations or editing of the splice sites. So if you go back here, if this site is mutated here or has a protein bound to it or something or another, it won't be recognized. And what the cell then does, it takes the one that's further downstream and skips this entire exon. So if you can't see, if the cell can't see this splice site here, let's say this little, on the little orange part or reddish part here, if this splice site isn't working, it will just take the next one, and then you get this green to purple connection. Okay, so it's usually that the splice site is changed, and that's why we can't have the same. That's why we can't recognize a certain exon, and then the exon is skipped. Does that answer it? Okay. Okay, so which after RNA splicing, we'll go to RNA decay. So what happens to the RNA after it is being made and it is being exported into the cytosol? So there's a specific pore complex in the cytosol, in the nuclear pore, in the membrane that lets the RNA go through, and then it's in the cytosol. And after it's done its job, so it gets translated as into a protein, which is a whole nother lecture. After it's done with its lifetime, it gets degraded. Okay, so RNA processing occurs while transcription is ongoing. So again, we tend to think linear, okay? We have your whole pre-mRNA transcript, and then after it's completely made and it's off the DNA and off the RNA polymerase, it gets processed further. That's again not the case. So while the RNA is being made, as soon as the 5' cap is out or the 5' end is out, it gets capped. Okay, so we don't wait until the whole thing is made. We can also splice out introns while the RNA is still being made. The only thing that we can't do before the RNA is completed is add the poly-A tail. For that, we have to wait until the RNA is completed. So that's just something to remember, that things happen all at the same time in the cell. The RNA is then exported into the cytoplasm, where it can then be translated. The export happens after processing. So only RNAs that don't have introns anymore will be exported. Only RNAs that are capped and polyadenylated will be exported. For the RNA export, we require specific proteins. And what I want you to remember is that we need proteins that bind both to the poly-A tail and to the 5' cap for the RNA to be exported, which also is again another safeguard from taking out RNA that's not processed yet. Okay, all right. Okay, this is the nuclear pore. You don't need to memorize this structure. I just want you to remember that it's a huge complex that's necessary for the RNA to be exported. Okay, it's not just RNA wiggles its way out. It requires this huge nuclear pore complex that recognizes the cap binding protein and the poly-A binding protein. It also recognizes a little transport receptor that binds to the RNA, and then only when everything's together does it get actively exported into the cytosol. So it's a huge complex that's required. Okay. This is the same. We recognize the poly-A binding protein. We recognize the cap, the transport receptor. The RNA gets exported, and then we initiate protein translation. Okay, so it's important to remember we do have poly-A binding proteins in the nucleus. We also have poly-A binding proteins in the cytosol. They're not the same, but it doesn't really matter. Just remember that we need those poly-A binding proteins in order to protect the 3' end of the RNA. Okay, which then RNA decay? How do we get rid of the RNA? There are many more pathways here than I will teach you now. But in general, we have decay that starts from the 5' end, and we have decay that starts from the 3' end. We also have decay that starts in the middle. Okay, so you can start anywhere, but you will need specific enzymes and mechanisms. So it doesn't just happen randomly. So for the 5' end to be decayed, you need to decap the 5' end first. It's a specific enzyme that's only active under specific circumstances. Once it's decapped, it just gets degraded. Okay, so once the protection's gone, there's no more stopping it. The same with the poly-A tail. First, you need to deadenylate it, which is a specific enzyme, which can't work if there's poly-A binding proteins. But once you pass that poly-A tail and that's gone, it gets degraded very quickly. So the regulation happens at the initial steps of getting it degraded. There are more RNA decay pathways than that, and eventually in the next probably year or two, you learn about microRNAs and how microRNAs can bind the really short RNAs. They can bind to an mRNA, and they're complementary to it. And when they bind to it, the mRNA gets cleaved in the middle. And when it gets cleaved in the middle, you basically have an open 3' and 5' end on the two sides, and then the RNA gets degraded. There are also other decay pathways. So there's been several Nobel Prizes for RNA decay and how it works in the past 5 years. So it's an ongoing field of research, and I'm sure we'll learn more about it the more we learn about RNAs. Yep. Yes. Yeah. The decapping enzymes, or which one? Okay, so the poly-A binding proteins. Okay, so the question is, what do you need to know? You need to know decapping happens on the one end and has to happen before decay. And the 3' end, you need to deadenylate before you get that kind of decay going. Kind of what's in this slide, okay? Decapping, deadenylase, the two pathways can go together. The other question was what happens to the poly-A binding proteins that protect the poly-A tail. So when we say something binds to an RNA, and even if it binds really strongly, it doesn't mean it's always there. Proteins and protein RNA interactions, protein DNA interactions, always kind of cycle on and off. If a protein is actively translated, they cycle on and off even more. So let's say in 90% of the time, a poly-A binding protein is bound to the very end of the tail, which means most of the time it's protected and it won't get quickly degraded because the exonuclease has to find it. Sometimes the exonuclease will find this little section and take a little bit of the poly-A tail away until it comes to the next poly-A binding protein. So there's many, a 300 base poly-A tail will have 10, 20 poly-A binding proteins bound to it. So it's just by chance that, it gets shorter and shorter over time. It's a on-off cycle system. So any repressor you have bound to your DNA, even if it's a super tight repressor, it won't always be there, okay? The better it binds, the more often it will be there, but just biochemically, it will cycle on and off. Make sense? Good, but really for the exam, poly-A binding protein protects the tail, but it can't indefinitely protect it. Okay, so finally we get to RNA modifications, and I think you'll probably get out a few minutes early. RNA modifications are other things that can happen to the RNA after it's being made. So we talked about mRNAs. There's also rRNAs, which are ribosomal RNAs, which are the RNA components needed for the ribosome, which is needed for translation. These ones are not capped and not adenylated. So in theory, they're not protected from degradation at all. It's interesting, they get made from one precursor RNA that just gets cut into different sections that then assemble to a bigger ribosomal complex. So, so they are somewhat the exception to the no operon rule, because with one pre-mRNA, you have different RNA products in there. But operons, we always refer to protein coding genes. Ribosomal RNAs are not protein coding, so it's still not really an operon. Okay, but so the ribosomal RNAs are being made as one RNA chopped into pieces. So the way how these RNAs are protected from degradation is that they bound to proteins. So the same with this, I just said the spliceosome has RNA components. They also don't have a poly A tail. They also don't have a cap. So they assemble into those huge complexes with proteins, which is what then protects them from degradation. Okay, because also once they are assembled, so they fold into their funky three-dimensional structure. Once they assemble, they don't need to be opened up again. So mRNAs that get translated into proteins, they need to be open for translation. So they have to be somewhat linear. Whereas those ribosomal RNAs, they can form into a tertiary structure, can stay there, be bound to proteins, so they won't get degraded. Yep, question? Oh, okay. So the no operon rule. I had mentioned before eukaryotes don't have operons, right? Operons. They have their one RNA or one gene being made into one protein, basically, except for the splicing thing. But we don't have the 3 protein products in one gene as we had in bacteria. The lac operon has 3 proteins that it codes for. So this is, you could technically say this is an operon. Okay, because you have 3 different components in one pre-mRNA. But since they're not protein coding, they're still not considered an operon, right? But they are 3 functional units within one gene. Good? Okay. Okay, so another thing we can do is we can modify bases. And this doesn't really happen in mRNAs because mRNAs are supposed to be read into proteins, so they have to stay true to their nature so that they the translation machinery can read them out. But if you have non-coding RNAs, you can modify bases. So you can make a uridine into what's called a pseudouridine. Okay, so this happens, modifications of bases largely happens in non-coding genes. Okay. tRNAs are highly modified, rRNAs are highly modified. The other advantage it gives them is that they again not recognized by nucleases very well. Okay, as soon as you don't have your regular bases anymore, the exonucleases that chop down RNA don't work as well anymore. So these are some modified nucleotides. So now, again, please don't memorize the structures, okay? Try to remember that we can modify nucleotides and they can change their function, okay? It can change how they act, how they form tertiary and secondary structures, how they interact with proteins, and how they're being recognized by proteins. Okay, so that's the main take-home message. And then non-coding RNAs tend to be highly modified. If you want to, memorize the structure, if you're going to go into chemistry. So if you ever go into, I don't know if any of you are going to be chemists. Chemists will have to know these structures later. Biochemists do not. Okay, which gets us to the end of today's lecture. We'll have capping, polyadenylation, splicing, export, degradation, and nucleotide modification. And with that, I'll let you go early. Enjoy the rest of your day.